Hello. Oh, and you started the recording for me. Thank you. That was you, Ann?
No.
Oh, that was Ken. Great. Okay. Thanks, Ken. So our program today is Feathered Palettes, the Science of Bird Colors with Dr. Jenna McCullough. And Dr. Jenna McCullough will take us behind the science of feather coloration, from pigments that turn northern flicker feathers red or yellow to the surprising mechanisms that create pure white plumage. She earned a dual bachelor's degrees in biology and wildlife resources at the University of Idaho, and then completed her master's of science and PhD at the University of New Mexico, where she studied kingfisher evolution, and has now recently moved to Los Angeles to begin a National Science Foundation postdoctoral fellowship at UCLA, and also with the Natural History Museum of Los Angeles County, where she'll be studying how birds produce white plumage. So without further ado, I'll let her take it from here.
Great.
And
I can
switch
see
over.
in there. Yeah.
Yeah, okay. And
Oh,
now I'm gonna
perfect.
remove our podium and face you to the audience here so that you can see our expressions, etc.
Great. Well, thank you for the
Oh, that's not
introduction.
good. It'll be back
Are you ready?
with a new lock.
Well,
Okay, well, I gotta redo this sort. 1 second. Technical difficulties. There we go.
Okay.
it's okay.
Oh, could you mute yourself? Because I can hear myself through.
Oh, okay, our feedback was cut off, so I thought it was fine.
All right. Now with a thumbs up, can you hear me okay? Great. Awesome. Well, hello, everyone. Thank you for having me. I'm really excited to talk about feather color evolution. As Kanshu mentioned, I'm a postdoctoral fellow at both the LA County Natural History Museum and the Ecology and Evolutionary Department at UCLA. So I'm really excited to talk to you all about some recent research on bird coloration, including some of the work that I'll be doing for the next few years. So let's see. Okay, so before I get into the science, I often get questions about how I came to have a career studying bird evolution. And I first wanted to start off by briefly explaining how I got where I am. Let's see. And I like to joke that I was actually born to love birds. So I am the third generation of female bird watchers in my family. My mother, who is watching on Zoom, and my late grandmother were both lifelong bird watchers. And the roots of which are actually in California. So my grandmother lived in San Diego for decades, working as a docent for the San Diego Natural History Museum. She and her friends created their own birdwatching group that was active for 40 years in the area called Tweet Meet. And in the bottom right here is a picture of their group. They had found a great horned owl while birding, and they paid for it to be taxidermied and donated to the Natural History Museum. And so her claim to fame, my grandmother's claim to fame, is finding the first record of golden-winged warbler for San Diego County in her backyard in 1984. And so she inspired my mom to love birds, and in turn, my mom raised me alongside a pair of binoculars. And so I'm originally from Boise, Idaho, where my mom was constantly taking me outdoors or camping or going to the zoo. And my love of animals was really reflected in my choice of childhood Halloween costumes, which ironically represent two sides of the contentious issue between birds versus outdoor cats. And my first eBird species is actually kind of funny. It's a snowy owl that my mom hoodwinked me into seeing when I was a teenager. And so I knew I wanted to study animals. And when I was 18, I went to the University of Idaho, where I began working on a number of bird research projects relating to, the breeding biology of owls and sage grouse, as well as volunteering at bird banding stations. And after 4 years of research and two bachelor's degrees, I applied to graduate school and got rejected from every school I applied to. So instead, I went to Columbia, where I worked on a project studying the breeding biology of Andean birds. And what that really meant is that I got to hike around the cloud forest all day trying to find bird nests, which was a really fabulous experience and really exposed me to the diversity of birds in the tropics. And then afterwards, I volunteered at the Burke Museum in Seattle for several months learning how to do museum specimen preparation, which is where I really cut my teeth on museum science. And so then in my second attempt at graduate school, I went on to do a master's and then a PhD at the University of New Mexico, where, as mentioned before, I was working on kingfishers and their allies, Crassiformes. And so during my graduate career, I had the opportunity to do field work in a remote island archipelago called the Solomon Islands to study birds in the wild, and also traveled to a bunch of different natural history collections around the world to study bird specimens collected over the last 150 years. And in between, fitting in birdwatching trips to the American and Asian tropics, as well as Africa. And so after I finished my PhD in 2024, I started a postdoc at the University of Kentucky. And so for those who do not know, a postdoc is meant to expand your research background while you search for the coveted tenure track job, which is the position I am doing right now. And so just in the last month, I moved back across the country to start a National Science Foundation fellowship with UCLA and the Natural History Museum of LA County to study feather coloration. Now, I'll be talking about that later in the presentation. So, why study color? I think that I am not unique to say that when I think about the 11,000 species of birds on this planet, one of the first things that come to mind is their amazing array of colors. And feather coloration is important in all aspects of a bird's survival and reproduction. And I'm interested in how and why feathers' colors change to produce what we see in nature. And it's important to understand how different colors are produced. So bird coloration is a complex hierarchy produced by these integrated modules that work together, these integrated structures. And what we see is not what birds see. All the colors that we see are a result of 3 color cones in our eyes. Birds have a 4th color cone, which means they're sensitive to UV light, and they perceive a wider range of colors than we do. And so in the past, to study bird colors, ornithologists have had to use these color swatches to be able to name the colors that they are observing. But this is obviously subjective, and it's just a fraction of what the birds are perceiving themselves. Now, we can now measure feather colors with a thing called a spectrophotometer, which can quantify what wavelengths are being reflected, meaning higher up on this graph, and which are being absorbed, lower on this graph. So we can also break color down into where different colors occur on the bird's body to create the arrangement of patterning. And we can even look at differences within a single feather, which is called macrostructure. For a single feather, there are different regions. The plumaceous portion of the feather, also called the downy portion, is important for keeping birds warm, but it's often hidden. And what we see when we look at a bird is often the pennaceous portion of the feather. And we have the rachis, which is like the trunk of a feather, and these different branches that come off that are called barbs. And zooming into a feather, we can look at the microstructure, which focuses on these barbs. So barbs are comprised of a smaller trunk called a ramus, and it too has these smaller branches called barbules. And the organization of these barbules, or the thickness of the ramus, they all contribute to the overall color and are different things that ornithologists can measure. And we can even go smaller inside the barbs to see how things are arranged internally, and that is called nanostructure. How light wavelengths are scattered or absorbed is very important for structural colors, and that relies on the nanostructure. So this could be iridescence, like a hummingbird's gorget, or non-iridescence, like the blues, greens, violets, and UV colors. And where in the feather barbs these structures occur that scatter light also differ. So for iridescence, the important structures are inside the barbules, whereas for non-iridescent colors, the structures are in the barb ramus. And when we look inside of that ramus, this is a SEM picture of it being cross-sectioned. What we can see is a spongy matrix that is refracting specific light wavelengths. So as light goes into a feather, like in this case for this blue feather, the blue light wavelengths are being scattered and refracted back to the viewer, and everything else is being absorbed by this black melanin layer. Iridescence is similar, but it occurs in the barbules. So here, light is being refracted off of this melanin layer, which is causing disordered scattering, causing this kind of glossy, oily look. And in things like hummingbirds, there are finely organized layers of melanin that causes different light wavelengths to be scattered at different viewing angles. The important thing to note from this is that nanostructures can be complex and that it relies on the actual structures that can be seen with a microscope to cause this different light refraction. And so nanostructures are focused on scattering light, but we can get even more detailed by studying the molecules that absorb color. So birds use chemicals called pigments to absorb specific wavelengths of light. And we can break these up into two categories, melanins and carotenoids. Melanins produce blacks, browns, and rufous colors, and carotenoids produce reds, oranges, and yellow colors. And most carotenoids are sourced through the bird's diet. And so here, a pigment that produces red is caused by absorbing all the other light wavelengths except red. So I'm going to talk about these three stories related to feather color research. The first will focus on pigmentary color evolution in flickers. The second will be on microstructure differences in tanagers. And then I'm going to talk about my research on nanostructural evolution of white colors. And so to begin, this first story was led by one of my postdoctoral advisors, Dr. Stephanie Aghion at UCLA. She studies color evolution in northern flickers, which are famous for having two subspecies defined by the color of their wings and tails, either being red shafted or yellow shafted. But there's also different other traits that differentiate these two subspecies, particularly their malar stripe and their throat and different head colors. And so red shafted flickers are found in the west, and the east has yellow shafted birds. And this orange section is where they hybridize. And these hybrids have orange shafts, but also a variety of traits related to their head. And in the hybrid zone, you can see yellow and red shafted birds side by side, along with, obviously, hybrids. And so Stephanie wanted to study the genetics of this hybrid zone and worked with the Cornell Museum of Vertebrates to use both historical and modern specimens along this transect that goes through Colorado and Nebraska to see how the birds on both sides of the hybrid zone and look at their genetics. And so hybrids can have a range of different traits from the colors of their wings to also different portions of their head. And we can use these hybrids to study how genetics differ between hybrids and non-hybrids. So Stephanie sequenced about 70 birds spanning those in and out of the hybrid zone and compared their genomes to look at differences in the genetic code that could explain differences in their plumages. And so these markers are called single nucleotide polymorphisms, which are also known as SNPs. When comparing these markers across the genomes, we use something that's called a Manhattan plot. So points that are higher mean that they are more different at that site, and lower means they're more similar. And so when scanning across the genome, you can see regions that rise like skyscrapers to tell us there's a lot of genetic changes there. So when she compared the red and yellow shafted individuals outside of the hybrid zone, she found that the average, which is this red line at the bottom here, showed that they were very similar. There was a lot of different peaks, a lot of different skyscrapers here. But when she just focused on hybrids and included information about these different color traits, she found that there was only a few differentiated regions across the genome that are associated with color differences. And these all had to do with color genes. So these included some of these genes. You don't need to worry about their names, but it also included carotenoid genes. Now, what's really exciting is that this one that's bolded is well known to cause a switch between yellow and red pigments. So this is really exciting because it explains the differences in the red and yellows of the wings and tails of these birds. One gene. What's also really interesting is that the melanin genes were also associated with changes in the carotenoid-based traits, including the malar stripe, the neutral patch, and the color of the wings and tail. So just in the same way that different parts of the bird feathers interact, like that hierarchy I explained before, and that they're interacting to produce color, so are different types of genes, both melanin and carotenoid genes, to produce a suite of colorful traits. So with hybridizing flickers, we've learned that there are only a few differentiated regions of the genome that produce color differences. The next story focuses on the microstructure of tanager feathers. So this section focuses on a study that was published last month in Science Advances, led by postdoc Rosalind Price Waldman and her advisor Cassie Stoddard at Princeton, as well as my other postdoctoral advisor, Allison Schultz at the LA County Natural History Museum. And so this group has been studying tanagers for a long time, and they are very interested in how tanagers produce the amazing arrays of colors that we see. And so for this project, they were focusing on measuring color with a spectrophotometer, as well as taking different measurements of individual feathers. And while they were doing this research, they were looking at these individual feathers and noticed a pattern of this interesting kind of black spot here. Not the bright green color or the plumaceous part, but kind of that little black spot. And this black spot is completely hidden when the feathers are normally arranged. Then they realized that this was different for carotenoid colored feathers, which also had a white spot. And so anything that was structurally produced had a black spot. Anything that was carotenoid produced had a white spot. And so they called these spots the achromatic layer. And when you think about the colors of a bird, this means that there are hidden achromatic layers that we never see and have never been studied before that we don't even know affects the color of the more colorful tips of the feather, the pennaceous part. So naturally, they wanted to know if these achromatic layers really do affect what we observe overall for color of these patches. So they measured the colored portions of the feathers on different backgrounds, either white or black. And then they found that yes, background color did have an effect for how bright the colors were. So this is one, that, I'm not sure you can see my mouse. This is a carotenoid feather that has a normal white background, is very bright. But when you put that same feather on a black background, it lowers in brightness. And structural colors do show differences both in a bit of the shape of this curve, and then also changing their overall lightness. So when feathers, again, were mismatched to their normal achromatic layer, they changed their overall color, just by how we're viewing it. So let's see. So they did this for about a dozen tanager species, all confirming the same results. Now, these were on backgrounds, solid white or solid black, that don't exist in nature. So they took it a step further and redid their analyses, but using actual achromatic layers of a specimen. So on this one specimen, they trimmed off the colorful portions of feathers to expose the achromatic layers for two patches. One on the head that had the black achromatic layer, and one on the rump that had a white achromatic layer. And they did the same measurements, and they confirmed these same results. So yes, achromatic layers are very important for overall color. They also looked at whether this could explain differences in plumage colors between the sexes, called sexual dichromatism. So in a lot of sexually dichromatic tanagers, males and females have the same plumage patterns, but males are more vibrant than females. And that's because males have larger achromatic regions than the females. So in different patches, males were brighter than females, and they also had these larger achromatic regions. So here's a female here. You can't really see much white here, maybe a little bit. And then you look to the male feather, and you can really see this defined achromatic layer on that feather. So with more of the appropriate achromatic layer, so white for carotenoids, black for structural base colors, these colors are more vibrant. And what's really cool is that when they measured those same feathers, but on the wrong background, so a carotenoid-based color on a black background or a structural-based color on a white background, they found that the males were no longer brighter than the females, meaning that the differences between the color differences between the sexes were erased just by changing the background. So these findings are really interesting. And what's more exciting for someone who, thinks about kingfishers and non-passerines, is that they found these achromatic layers across different groups of songbirds, not just tanagers. So I think it's opened up a really huge avenue for future research on plumage color evolution, because now we need to be thinking about not just what we can see, but what's just underneath the surface of these feathers, that hidden achromatic layer contributing to the differences in color. So with tanagers, we've learned that there are these hidden achromatic layers of feathers that influence the colors that we observe, and that can also explain differences in sexual dichromatism. And so my last story focuses on my own research on white coloration. And so for a chapter of my PhD, I was interested in color evolution for a genus of Indo-Pacific kingfishers called Todirampus. And so this is a genus of around 29 species that all either have non-iridescent blues, greens, or violet backs with white or rufous bellies. But I'm actually not going to talk about the results of that particular research. I wanted to actually tell you about a story that kind of was a result of just working with specimens in natural history collections. And so I was particularly interested in wing shape and just general color measurements, not taking feather samples for over 1,000 Todirampus specimens. So I measured 1,000 Todirampus specimens, their wings and their color, from museums across the US, Europe, and Asia. And as I was pulling these 1,000 specimens from cabinets, I'd often wear a headlamp because I really don't like working in dim light. And in 2022, I was going through the drawers at the American Museum in New York City, and I noticed that the specimens at my eye level had what I thought was an optical illusion because their white bellies were reflecting these green colors back at me from my headlamp. So here's a video. It's not always viewable on some projectors, but you can see this really light green sheen on the feathers. And so usually something that is white is always white, no matter what angle the light is or your viewing angle. It will always be white. So this is really weird. And obviously I kind of thought it was crazy and I needed a break from staring at so many specimens for so many hours. And at first it was just a green sheen. But then I noticed that there were species that had violet backs with the same violet sheens on their white bellies. So here's another video where just by changing the angle of the viewing and the light, you can change the color of their belly from white to a different color. And so when I had discovered this, I didn't really know how to describe it. So I called them washes, and that's just how I'm going to describe it for the rest of this presentation. So in every case, the wash color matches a structurally produced color found elsewhere on the bird's body. So for Todirampus kingfishers, the matching structural color was on their backs. So violet back birds had a violet sheen on their belly, green back birds had a green wash on their belly, and blue back birds had a blue wash on their white belly. And so here's what it looks like on a compound microscope with angled light. So you have these white translucent ramus, and then as you go out to the tips of the feathers, you can see that they begin to change color. and have this like greeny blue color here. So this effect disappears when you have the light just overhead. You can only see it really at an angle. So again, what we have are these white-bellied birds that look white in normal lighting conditions, but with a bright angled light in a dark environment, these bellies reflect a different specific color that can be captured with a spectrophotometer. So for this middle bird here, it's an ultramarine kingfisher. It's found on the it's found in Vanuatu, which is kind of near Fiji. It has a spectral reflectance profile for its violet back that has a peak in the violet wavelengths, violet and UV wavelengths, and then it absorbs a lot of other wavelengths here. And measurements from its chin show a profile typically associated with white, meaning that there's many wavelengths that are being reflected equally with this asymptotic line. But the breast, which shows this violet wash, is somewhat a mix of both. It has a peak in the same wavelengths as purple, but it still reflects a lot of other wavelengths like a typical white feather. And so the same pattern is evident for beach kingfisher, which has this green wash and this little small peak right here. So what this suggests is that there are maybe ordered nanostructures that are producing this effect. So as I had mentioned before, non-iridescent colors, they have their spongy matrix inside of the barb, inside of the ramus. But they also have a melanin layer that absorbs light. And they have, again, the organized spongy medullary matrix that reflects specific wavelengths. A normal white feather is thought to have unordered nanostructures, so no spongy matrix, and lack melanin. But white actually hasn't really been studied that much in birds because they thought it was pretty boring, actually. And so because of these washes seem to have a mix of both, it suggested to us that this effect was through the same ordered nanostructures that produce the purples, blues, and green colors, but that lack melanin. So what does this look like? So here is a violet colored back feather from the ultramarine kingfisher. It has barbs, the ramus, that are violet with dark barbules branching off of them. And as expected, when we look at a cross section, you can see a organized spongy matrix. It literally looks like a little sponge. And it also shows melanin, which are comprised in things called melanosomes. These little rod-shaped things. And then chin feathers have translucent ramus and translucent barbules. So cross sections of that show a lack of melanin and no matrix, again, what we expect. But the wash feather is, again, a mix of both. The barbs here are this light violet color with translucent barbules. And then this transverse cross-section confirms the presence of a spongy matrix, but without melanin. And so the same is true for the beach kingfisher and its green wash. The green feather has a typical spongy matrix and melanin, and the wash feather has a spongy matrix and lacks melanin. And so when you zoom in further, the spongy matrix looks largely the same between the two, which to me is pretty wild because when you hold these two feathers and look at them with your bare eye, one is green and one is white. But when you look at them microscopically, the only difference is that the green feather has melanin and the wash feather does not. Now, it's only been in the last 10 years that we've known about the existence of white being produced with these ordered matrices inside of feather barbs. So these ordered, like this, the spongy matrix. And out of 11,000 species of birds now recognized by the International Ornithological Checklist, we only know of this occurring in 3 species. But I observed these washes in nearly every single species within the genus I studied, Todirampus, across other kingfishers, and then across other families in their order. And now I've confirmed its existence in at least 23 different bird families, including tanagers, but also in groups like jays and bangas. But this effect has been noted in leucistic birds, specifically this leucistic Steller's jay in 2006. And so what this study showed is that the only difference between the white tail feather with a faint blue overtone of the leucistic jay and a tail feather of a normally colored jay was the lack of melanin. Both feathers still had spongy matrix. So to confirm this in species that aren't leucistic is really exciting and really interesting. And again, I think there's a lot to be discovered by studying white feathers. And so finally, we've learned that there's a lot to be learned from white plumages, and that not all whites are really the same. So I went a little bit fast, but sorry. This presentation was a lot of work by other people, actually. So by Allison and Stephanie, Rosalind, and all these things have already been published. And of course, everything that I presented today would not have been possible without natural history collections and the many, many people who have dedicated their lives to preserving, collecting, and caring for specimens in natural history collections, which truly are our libraries of the world's biodiversity. And I am also funded directly by the National Science Foundation, which funded me. So thank you for that. And so with that, I have plenty of time for questions. I'm assuming you guys are clapping. I can't hear you, but I can hear it in my mind. I think you do need to probably unmute yourself now.
Yeah.
Okay.
I'll repeat the questions for you if necessary. You can let me know if you hear them or not.
Yeah.
Yeah, go right ahead. And I'll raise your hand if you want.
What, my goodness. So what's your do you have a next goal you're working on in your research?
Yeah, so the last portion was really the beginning steps of studying white color. There's, as I said, there had only been a couple instances of white being produced with like a spongy matrix ever. And so I have found it in about 100 different species. So kind of looking at whether or not the ways that they produce white are the same. What is the genetics of, uh, of producing a spongy matrix? Or is it related to differences in melanogenesis, in melanin production? So there's been a lot of research looking at how melanin deposition, melanin production can change bird colors really fast. And so this is really kind of maybe the turning off of melanin production that might have like a green feather randomly, quickly through evolutionary time turn white. And with a simple change of decreasing melanin production. So looking at that. And looking at whether or not there's any functional changes. So when we think about melanin, one of its benefits is that it has UV, can protect species against UV damage. So there have, like with birds with black on their head, if it like randomly turns white, they've lost a layer of UV protection. So looking at that and other aspects of functional differences in white.
Question.
Yeah.
So do the white feathers with the spongy matrix, we hear a lot about how birds will show different colors of their feathers in like ultraviolet. So do they show up different in ultraviolet than white feathers that don't have the spongy matrix? Or like, can you comment on maybe how birds perceive these different structural white feathers versus the white feathers with the wash? I don't know if that made any sense.
No, it makes sense. I think you might be thinking of, like puffins when you shine a black light on them, different parts of their bill will like fluoresce. Um, that doesn't occur to my knowledge with these special white plumages. We can see that there are, peaks, the graphs I was showing where there was a little peak in the purple and the violet colors. That's a little bit in the UV spectrum, but uh, I think that birds can definitely perceive these differences just like we can. It's just, I think in this case, we just have not noticed it um, to study it. But I definitely think that they probably can be observed because birds have a wider range of their ability to perceive color than humans. So, but is it like only in UV color that this is occurring? I don't know. Like I have yet to find a white feather that has a peak only like that asymptote that I was showing, like a typical white that only had a peak in the UV. But that's also because the way I'm observing these special whites, the way I found them is literally going with a headlamp and a collection and looking at birds with my headlamp to see if I can see any differences. It's just been through my own vision to see them and identify it.
This is a question.
Oh, we have a question from Ian Taylor. Is it energetically costly for birds to produce the spongy matrix?
I'm imagining that there could be a cost. I mean, it requires keratin. So spongy matrices are really just keratin and air. I don't think it is the same cost as producing a carotenoid pigment, which is sourced through a bird's diet. Um, but the the actual energetic cost, I don't know. Obviously, there's something extra that's being done. The one of the hypotheses is that there could be some kind of constraint where uh, it's like I'm losing my train of thought. Where it's just because it might not be as costly as other types of color. Kind of lost my train of thought there. Anyway, sorry if I didn't answer that all the way. Who
And
knows? I don't know yet.
I have actually a similar question, if that's all right.
Yeah.
There's so when we think about like coloration, especially like carotenoid coloration in birds, I feel like people want to call them sort of like measures of quality or fitness or something. But if really the
Mhm.
brightness of the carotenoid coloration, at least between the sexes, is because of the melanins, do you think that individual variation between males is also because of melanins, or do you think that, and like really it's just melanin production that is the thing that makes us think that they're brighter or higher quality or whatever? Um, or is it just between the sexes?
Are you talking about like the tanager portion? Like the achromatic layer, or I guess
Yeah,
I
the
was,
yeah, the
yeah.
achromatic white or black layers. So, or in the case of the carotenoid ones, the white layers.
Hmm.
Uh, like, is it the ability to produce a really nice white layer that we should be thinking about as like as the thing that makes really bright males, or is there still a big carotenoid like density or something?
You know, I think that could be a follow-up question for that study, as to whether or not, like, if you like a trade-off, like if you have less carotenoid pigments because let's just say you aren't getting enough of the food that you need, but you have evolved to have sorry, that's my cat, um, a larger white achromatic layer, would that change the sorry, would that change The overall color, I'm not sure. I think, when we think about structural colors, that has really not much to do with with signaling on like because carotenoids are based off of a bird's diet, whereas structural colors are not. So I'm not really sure. It's an idea. I mean, this is like such brand new, like no one has ever really thought about achromatic layers, and there's still a lot of questions.
Yeah, okay, interesting.
Yeah.
Thank you. Other questions from the audience, or let me check the Zoom chat. Very neat stuff. Oh, I was also wondering about with the flickers, how do you get the intermediate orange flickers if it's just one gene?
So there could be, if there's a male red shafted and a female yellow shafted, it would be the potentially not enough uh, of the gene is being, or not enough the proteins are being produced caused by that gene to produce a fully red or a fully red flicker. So it kind of comes off as orange by producing not enough. Because
Oh,
the way that that gene works is that and it starts yellow and it initiates another enzymatic reaction to cause the red color. So if you don't have enough of the enzyme to produce that reaction, you might just not end up fully red. It's just more like orange.
okay, interesting. Thank you. All right, well, I suppose if there's no other questions, then that's it for our presentation this evening. Thank you all for coming in person and in Zoom. We have one last question from Michael.
On the,
Oh,
on the,
thanks.
on the intermediate flicker, on the mustache, the male stripe. Sometimes you get like both a red and a black color in the stripe. I swear I've seen some that looked, I almost would have called it, violet or something like that, some combination of red and black. Does that happen? That there's both colors from both of the parents in the A stripe, yellow stripe?
I'm not sure. Uh, uh, Stephanie would be a great person to ask. But yeah, I'm not sure. Sometimes when the reds get a little bit darker because there could be more melanin production, sometimes it kind of, gives off an interesting view in the field. It doesn't look how you'd expect. But she also studies aberrant bird plumages too. So like when a bird has a inability to produce melanin, you can see these really crazy ranges of different plumage traits. And so studying something like that. But that has nothing to do with what you specifically asked, and that's because I don't know.
So it's possible that both colors, have both the red and the black, or that there's just more. I'm not sure. Dark.
Yeah. If there's both carotenoid pigments and also melanin, it would probably, just make it like a really dark plum. Maybe it might look like a darker
I'll go.
reddish.
All right.
Can I ask a question?
Yes, yes.
Yeah, just, let me try to get the phrasing right. Is the dressing of the male, was that substantiated to facilitate an attraction to the female for mating purposes? Or is this a feature of sort of the ecology around the weather, if you will, to sort of protect its skin from sunlight, the weather?
Mhm.
Would you like me
I
to
think,
question, or did you?
so what I understood was that whether or not it was driven by maybe mate choice between males and or from females selecting different colored males versus it being a thermoregulation or UV protective type of adaptation, right? Is that,
That's I understood. Thank you.
yeah? Um, and so I would say that I think when it comes to these really bright colors, uh, I think many people would say that sexual selection might be driving color evolution within tanagers per se. But whether or not things like for the whites, um, when you lose melanin, like so for example, that beach kingfisher is in really bright environments, and it has like a lot of the kingfishers that I study, they have like green caps on their heads. And beach kingfisher has evolved to effectively lose its melanin production in its head, which is where there's a lot of UV radiation. Um, there's been some work that has looked at bird skin and has shown that some birds that have white feathers on their heads, and this is irrespective of the special white that I was talking about, they were just being like, oh, it's a white head, let's look at the skin. And so in some of those cases, there's melanin production in the skin to have that protective UV protective layer, and not in the feathers. So
All right.
sometimes, yeah.
Oh, so there is sometimes a protective or environmental
Yeah. That
effect.
there's, yeah, but when it comes to like the differences between the males and females in the tanagers that I was talking about, um, you know, I would place a bet on sexual selection or um, signaling between maybe for like territoriality between males.
Hmm. We have a question from Dale Val in the chat. He
Mhm.
said, I've read that birds of paradise are basically elaborately colored and feathered crows. Are crow feathers and bird of paradise feathers being compared to study what mechanisms are in play in the generation of the elaborate colored feathers?
So birds of paradise are in a larger group of birds called corvides. It is a large clade that includes things like vireos, includes vangas, um, it includes, shrikes and crows. So maybe that's what he meant by saying they're like crows. But they are on a different evolutionary branch than like true corvidae crows. Um, there has been a lot of work on their feather evolution. One example that I didn't explain was there are different, I can't remember the species, but this is a well-known species that the male, when it is attracting females, will move its feathers up to be this big black, like almost like a UFO with eyes, vivid green. I swear I can't remember its name. But the feathers, the black feathers that produce that, have specific adaptations to make them absorb even more light, and they're called super black feathers. Um, some of the crows that I have studied in the family Corvidae, the specifically like New World jays, they have the special white that I was talking about. But yes, there's a lot of people working on birds of paradise feathers. They're incredible.
Do scrub jays have it by any chance? Sometimes when you see a scrub jay belly, it has that little, that's not what that
Yeah,
is.
so it's not that. I've noticed it in you know how some Stellar's jays, there's differences in like the eyebrow color where it might be like I think it's black in Alaska. Some have white in the West, and then in Mexico it's blue. The differences between all of those could literally just be the amount of melanin production. So, like
Oh.
in Alaska, if there's just like there could be spongy matrix in the same patch of the eyebrow of a stellar jay, but there's so much melanin that it's absorbing out the signal of the spongy matrix. And then if it decreases its amount of melanin production, it might look blue, like in Mexico. And then in the West, where it's white, if it loses all of its melanin, the same structures are there to where it would look white, but it would have this little blue sheen to it. So also it occurs in blue jays. So if you've ever looked at a blue jay, its breast kind of looks like it's like white, but it's kind of like a dingy white. And then you look at its tertials on its wings, and they're really bright white, brilliant white. The whites are produced differently. So on the breast, it's like a typical white. And then on those tertial white patches, it's this special white. So it makes it look so much brighter. Yeah.
Thank you so much for presenting to
Yeah.
us this evening.
Yeah. Thank you for having me. Great. Well, have a nice day, everyone. And I hope you all get to see that incredibly rare bird. It's a little bit too far for me to drive.
Yes, and it has no
But yeah.
color whatsoever.
Ah,
Uh,
yes.
Yeah,
Great.
thanks again and good luck with your continuing research.
Great. Thank you. Bye.
Bye bye.
Oh.
I'm gonna end our